series. This morning, Jenny Mortimer is going to introduce our speaker, Kim Johnson. So I'll hand over to Jenny.
Okay, thank you, Rebecca. I hope you can hear me over the thunderstorm outside. I'm really excited to introduce Kim Johnson to give our talk today. So Kim is a senior research fellow at Trobe University. We can also call her award-winning Kim because she's had a host of awards for her teaching and research recently. And she works generally to understand how physical stresses, such as those generated by growth and wind, are sensed to initiate pathways that regulate the cell wall. And therefore, with that, how plants grow and develop. And so she is a CI in the ARC Medicinal Agriculture Hub, and she's previously worked in the COE on plant cell walls. And with that, I'll let Kim take us away.
Thank you very much, Jenny. And thanks so much for the invitation and for your patience after last time, the technical difficulties. Anyway, I'm in the office today, so hopefully all is well. And my talk today is called Don't Stress, Cell Walls to the Rescue, because cell walls are just the best. But I wanted to start off with asking you, I can get this to me, how do you feel? So I'm feeling pretty exhausted after lockdown, but that's not quite what I meant. I meant, how do you feel? How do you feel with your fingers? How do you feel heat? How do you feel spiky cactuses underneath your fingers in your skin? And I don't know if many of you noticed, but this year's Nobel Prize winners were the people who investigated how you feel, how people can feel these sensations. And we know that this is due to some amazing channels called piezo channels and other associated proteins, which are ion channels. So they're stretch activated, they stretch in these basal membranes under our skin, and they conduct electricity essentially, so calcium ions, and then these transmit to our nerve ends. So this is how we can feel the world around us and respond. But what about plants? I mean, plants are usually, superior organisms, in my opinion, to humans. So surely they must be able to feel as well. And they can. So this is a really interesting practice, which they do in Japan, which is called crop treading. So they go out and they stomp on their plants early in the season to make them respond to this physical touch, because they find that when they do that, they get a better yield. So if you look at the grain weight per plant, these treaded plants that have been stomped on actually produce more. And they think this is because they're giving them a bit of, hard love in the early stages and they toughen them up. But it does show that these plants respond to physical signals, and that can entirely change their growth. And we know this, right? We must know this because from the very beginning of a plant's life, as soon as it germinates out of that seed, and that very process of germinating, bursting out of that seed, and then growing through the soil and exposing itself then to the wind and the rain, these plants must have ways of responding to these physical signals. But also the very growth of a plant, it must take up that water, hydrate, and use that pressure. So because I've been doing lots of online teaching, I'm now going to ask you a question. So if Rebecca can do the first poll, please. Okay, so this is your first year biology test. Plant cells are hypertonic, isotonic, or hypotonic. Please put in your answers. So hopefully you're busily putting in there and you all remember first year biology. I have to say that my students did incredibly badly on this question. So let's see how you do. So Rebecca's in control, so she'll be able to tell when everyone's responded, or at least maybe 80% of you. It's as good as I could get in my classes. And then Rebecca, if you can share the answer.
Okay. Well, people aren't overly keen at responding. We only had about a 50% response. Okay,
that's probably about right for my classes.
And I can't respond as a panelist, sorry.
Okay.
So 67% are isotonic and 33% are hypertonic.
Okay, so thank you very much, everyone. Plants are actually hypotonic, so they need to take in that water to push themselves up against that cell wall so that they're really turgid, and then they can expand, use that for expansive growth. So hypotonic, okay, you can teach your kids that later. So they're under pressure, right? They're always under this pressure. So one thing people wanted to know was, well, where is this tension experienced? Because we have different shapes of organs. So we have these kind of cylinders of the stems and hypocotyl. We have the cotyledons and leaves, which are much more flat. So they experience these stresses in different ways depending on the tissue. But something that was really clearly shown was that the tissue under the most tension is the epidermis. And this was a really nice example of using a pectin mutant, so it can't adhere itself properly. And you see that these epidermal cells sort of peel away. And this integrity, this cell-cell adhesion in the epidermis is really important for creating that tension, as well as it's this kind of feedback loop. Okay, so it experiences the most tension, but is also part of causing that tension. And this is important for plants to be able to grow in their way and create all these amazing shapes. Okay, so it makes sense that the epidermal layer, being this outer layer, is under the most tension because it's being pushed up against by all these other underlying cells. And so that adhesion is really, really important. And it regulates growth and its interface with the environment. So it's kind of similar to us. You remember me talking about us feeling the physical sensations that when we touch. So it's kind of a parallel to these epithelial cells in our skin, and have this basement membrane, which is able to transmit these signals. Okay, so what's really important? You have to have that cell adhesion, as I said. You have to create this turgor, and it's all because of this amazing structure called the cell wall. So the cell wall is really, really important for these signals, these mechanical stress signals, and that integrity of these plants. And so it makes sense that you must be able to change that depending on the need of the plant. So again, I'm just going to test your knowledge here. Cell wall related people, just hold back a little bit, but I want to ask everyone else to just pop in the chat if it's open. Just remind yourself what that primary plant cell wall is made of. So feel free to pop a few answers in there. And cell wall people, if no one responds, feel free to go for it. Yay, thanks, Matt. Hooray. Excellent, thank you, lovely answers. Pectin, cellulose, really, really important parts. Bit of xylan, xyloglucans. So these major parts of the cell wall, these cellulose, we need those rigid cellulose, which creates this kind of steel beams that create this incredible strength. Pectin to allow that gel-like matrix for things to move around a bit. AGPs, thank you, Jenny, love you. Love AGPs, they're the best. Yeah, bit of protein, because we need to be able to modify this as well. And we need to be able to sense what is going on and change things when needed. Okay, so this is also kind of parallel to, I know there's a few people working on defense. So when you get, this is cell wall stress or cell wall damage, we get sort of similar responses because you need to modify that wall in response to these stresses. Okay, so how do we think this works? We have some kind of cell wall stress or some wall damage, and this could be that mechanical stress or other related stresses. But we must sense this. So the plants must have a way of sensing this and being able to respond appropriately. So these cell wall integrity sensors are really, really crucial to initiating these responses. Okay, then we get some signaling, and this could be directly, most likely to some cell wall components, but also to the nucleus, where we get these changes in gene expression and responses that include cell wall modification. And this must also be tightly linked to the cell's identity and cell-to-cell communication, because we need to maintain that integrity between cells and communication, so everything's responding together. Okay, so what could these cell wall sensors be? Okay, so we know that receptor-like kinases are really, really most likely candidates here, because they can they go from this cell wall, plasma membrane, cytoplasmic continuum, and they can signal. Right, and some of them are thought to interact with these cell wall components. But as I mentioned before, these stretch-activated calcium or ion channels, which are really well known in animals, are also likely to be involved. We know that there's calcium fluxes, but what's been really, really difficult, so like similar to those piezo channels that I mentioned earlier, where are they in plants? That's been really, really huge question. We just haven't been able to identify them. And to be honest, we have very little information about what interacts with the cell wall. These receptor-like kinases have been very hard to identify the ligands, and the complexity of these receptor-like kinases in plants is huge compared to other organisms, the animals. Okay, but one of the questions we were really interested in is these stretch-activated ion channels and what they were, the equivalents in plants. Okay. So that was a kind of simplified view. We know it's really complex. There's lots of signaling going on here that change the cell wall. So hormones, cell wall integrity pathways, and ROS signaling. So just to make it that I'm simplifying this dramatically, it is really complex. Okay, so what features though would we like to see in a mechanosensitive ion channel in plants? Well, it should be located at the plasma membrane and likely have many transmembrane domains because if it's responding to that plasma membrane stretch. Should allow the passage of calcium ions into the cell. It should be involved in epidermal integrity because, as I mentioned, this is the layer under the most tension. So this is where we need these sensors the most. Okay, and it should regulate the cell wall because that's part of these integrity responses. Okay, so I'm going to introduce defective kernel 1, DEK1, which was initially identified in maize as an epidermal mutant, and we have looked at it largely in Arabidopsis. So I worked with Professor Gwyneth Ingram in Edinburgh many years ago, and we were looking at this defective kernel 1 because of her interest in epidermal development. And long story short, eventually Gwyneth was able to undertake some, to identify that this is a mechanically activated calcium channel. Okay, all works with a rapidly mechanically activated calcium channel. So she did these patch clamp experiments where you patch a little bit of membrane and then you look at the transduction of ions. And she could see that in just normal Columbia wild type, you get this fast intake of calcium and this peters off when you stretch that membrane. But what was interesting that this is almost entirely absent in these defective kernel 1, DEK1 mutants. Okay, so this seems to be really important for these calcium channel ion activity in response to that physical stress. So DEK1 is an interesting, very interesting protein. So it has 21 to 24 transmembrane domains at this plasma membrane, and it has a calpain channel. So a calpain is a cysteine protease, but it's not a sort of degradatory protease, it's a regulatory protease. So these are involved in signaling pathways. They modify their target proteins, which are then involved in signaling. And this is fairly well known in animals. So there's a number of calpains been identified in animals that are involved in a whole range of processes, cellular processes. But what's interesting is that some of them are responsive to the calcium influx by piezo channels. So this looks like potentially DEK is this fusion of these piezo channels with a calpain domain in one protein. Aren't plants smart? Except for the fact that plants only have one, whereas in animals they have multiple. Okay, so located at the right place and allows the passage of calcium ions into the cell. So as I mentioned, it's involved in epidermal integrity. So I'll just show you a bit more detail about that. So we first looked at these embryo lethal mutants, essentially. So when you completely knock out DEK1, I mentioned that there's only one of them. So you knock it out, the plants are dead very early. So we looked next at some RNAi, but because we used here a 35S promoter, so it got it through that first bit of embryo development, but then essentially we created these skinless seedlings. They were very, very unhappy as well, but that also indicated that they're really, the the epidermis was really, really sensitive to loss of DEK1. Okay, then through a screen that Adrienne Roder was doing, looking for sepal mutants that lacked giant cells in the sepal, she identified a DEK1/4 allele. So a nice sort of moderately and mild allele that survives and we can have a look at. And we also have generated this miRNA DEK1 line, which is also quite mild. And these you can see have defects in their epidermal layer. So in the DEK1/4, they lack those elongated sepal cells. They also don't, as similar to the miRNA DEK1 lines, they don't create these interdigitated pavement cells in the same way as wild type, and they can also lose integrity between these cells. We also generated a calpain overexpression line. So this is just taking that active cysteine protease part and overexpressing it. And there we saw also a number of defects. And these plants are they will get really rumpled. They're a little bit squat and stunted. And they look in a way like they're being mechanically stressed all the time. And you get these additional cell layers and also defects in terms of cell division and expansion timing. But what we also noticed in these calpain overexpressor lines when we looked in the epidermis in more detail is that they get these thick epidermal walls. So it looks like, as I said, they're sort of just reinforcing that outer epidermal wall like it's being mechanically stressed. And this is much thicker than wild type in these miRNA DEK1 lines. And we saw also that there was changes in the composition of this wall as well. So they had more pectins. So as we know, the pectins are really important in these sort of stiffness of the walls and that adhesion between them. A student recently that I've had, Lazar Novakovic, he has been looking at AFM just to confirm that with cotyledon seedlings. So the AFM is essentially where you go along and press on the wall, and this is really in fine detail here. So he's looking at the outer, well, it's actually the inside of the epidermal cell wall, and he noticed that these are stiffer in those calpain overexpression lines, and you also get differences in the arrangement of cellulose. Okay, so looks like they're involved in epidermal, DEK1's involved in epidermal integrity, as we would expect of a mechanosensitive ion channel. And I've also started to indicate that it's involved in cell wall regulation. Lazar also looked at this in terms of cell wall damage. So you can put this, treat young plants with this component called isoxaben. So this essentially disrupts cellulose biosynthesis. And when you do that, so the ISO there, the Columbia plants, they just can't grow as well. And yep, they're not very happy. You get this swelling of these root cells and shorter roots. So this was exaggerated in the calpain overexpressor. So it looks like they're sort of hypersensitive to this cell wall stress. Whereas the DEK1/4, just pretty much insensitive to that cell wall damage. So this again looked like it could be involved in these cell wall integrity responses. And also another indicator of that is ectopic lignin deposition. And in both cases, strangely, but they were less able to deposit lignin in response to this cell wall stress. Okay, Lazar also looked at the movement of cellulose synthases, so these CSC complexes. And again, he noticed that there was modified dynamics of these CSAs in the calpain expression line. And this actually leads to lower levels of cellulose in these lines. Okay, so DEK1, we have identified what we think is the major player in terms of a plant mechanosensitive ion channel involved in these cell wall integrity responses. Okay, so the really big thing that we're missing though is, well, a few things. Sort of what type of stress this responds to. We'd really like to know how much stress is needed and what type of stresses that activates these channels and initiates these signaling responses, but also what are these substrates? So cysteine protease, we're still finding it really difficult to identify what the direct targets of DEK1 are that then initiates these changes in cell wall integrity as well as epidermal identity. Okay, so there's a few, I'm not covering everything here, but there's a few collaborations that I have on the dekl project. So with Aran Sampath-Kumar, so we co-supervised Lazar looking at this cellular mechanism. I still work with Gwyneth Ingram, so she's largely interested in seed and fruit development. With Malcolm Bennett on soil compaction, again, a really important physical process, and with Michael Leonard in biomass and more around flowering here. Someone's trying to call me and I'm just going to, it's about my computer. So I've been talking for quite a long time, so it's time for another poll. Rebecca, if you can please launch this next poll about who was the tallest person ever recorded. Fun fact again for you to take home. And the amount of people who put Andre the Giant will maybe age you. I feel like that's, you have to be of a certain age to know who Andre the Giant is. This one's much more low stakes, so you don't have to worry about if you get the answer wrong.
We have about 50% who have answered, so I think we're so I'll just share the results. There you go.
Excellent. So Sultan Kosin is currently the tallest person in the world, but he is not the tallest ever recorded. And you'd be safe to go with a Dutch name, but no, it's actually an American who was the giant of Illinois, Robert Wadlow, at 2.72 meters. And I put his dad there next to him to indicate the fact that he was enormous, like just, yeah, he's even taller than Matt Tucker, just a giant. And, you know, that's amazing for a human to get to, but unfortunately he did die at 22 because humans are not meant to be that tall. But plants, amazing, again, superior organism. Look how tall plants can get, 115 meters with these giant redwoods, making Robert Wadlow look like a little teeny tiny ant. How do plants reach this incredible heights? Well, it's all thanks to their wonderful cell walls again. And once again, once you get to that enormous height, you must be feeling an incredible amount of weight. So there's a lot of compression stress. So this stress, like, you know, weighing down on these plants, but also they can feel bending stresses as well. So you think about also branches, they're, you know, coming out at this angle, they must be able to, you know, they must reinforce themselves so they don't just collapse. And plants can reorientate back up again. I'm sure you've seen that where something's been blown over and then it can reorientate itself back up again. And these plants here on the coast, they've experienced that wind coming off the coast and they've changed their whole body shape to respond to those physical signals. So these secondary walls are incredible for providing the strength of plants to reach those enormous heights, but also are really crucial in terms of these responses to those compression and bending stresses. So once again, in the chat, just, you know, you've already said what's in a primary wall. So just remind me what is in the secondary wall, please. Yes, thank you, Scott. Lignin, definitely there in most cell walls, with the exception of things like cotton. Mannan, yay, you Jenny. You can put my favorite thing again there if you like, Jenny. AGPs, yay, definitely like to see AGPs. There is AGPs there, Matt. They're a minor component, but they're there. Okay, so in those cell walls, we have cellulose again, but it's more crystalline. Okay, so it has a higher degree of polymerization, which means it can get longer and can make thicker fibrils. Lignin often in these secondary walls that provide that rigidity and strength. Hemicelluloses that are a little bit different from those primary walls, and the wonderful AGPs. Okay, so the regulation of secondary walls, it must make that transition, right? So it goes on this transition from this primary wall to this secondary wall. And there's a lot of signals in that. So there's environmental signals that can initiate these, as well as just developmental timing. And it's controlled by these transcription factor cascades, starting with this NSTs and VNDs in the layer 1, and then cascading down to these MIBs, and then finally this synthesis of these secondary wall components, including cellulose, hemicellulases, and lignin. Okay, but is there a feedback from this? So there's been a bit of controversy about this. In primary walls, it's pretty clear that there is these cell wall sensing pathways, but in secondary walls, it's been a bit less clear, and really not many players have been identified. But we think this must occur because, as you can see, there is these responses to plants when they bend over and fall, they can reorientate. So we think there is some kind of responsiveness to the integrity of these walls to allow them to initiate responses. So what would we be looking for here? We'd look to see that cell wall sensors in secondary walls are involved in responses to mechanical stress. They should be expressed in these secondary walls so that they can initiate responses in those cells and cell walls, located at that wall membrane interface, similar to the mechanosensitive ion channels, and regulate cell walls. Okay, so I'm going to introduce you to some wonderful proteins called vesicle-like AGPs. So these are glycosylated proteins. So they have a protein backbone, which has all these sugars attached. And in the case of vesicle-like AGPs, they also have, which is represented here in this blue, a fasciculin domain, which is related to animal cell adhesion domains, and also a GPI anchor on many of them, which connects it to the outer leaflet of the plasma membrane. Okay, so these FLAs, for quite a long time, have been implicated in these secondary wall mechanosensitive responses. So in eucalyptus, it was shown that particular FLAs are expressed, for example, only in the lower level of a branch, and also in these, specifically in these secondary walls. In populus, they've been shown to be involved in tension wood formation. So you get these FLAs expressed when you get this tension wood, so when you get this bending, that helps reorientate these plants back upright, and it's a localized, so you only get it on one side. And when you lack this FLA6, then you have an impaired ability to respond and create this tension wood. And also in cotton fibre length and strength, FLAs have been shown to be involved in this process. So in Arabidopsis, there's 2 FLAs which were identified to be very specific to secondary wall development, and these are FLA11 and FLA12. And they've previously been shown when you have mutants that they're very brittle, and you can often get collapse of cells with secondary walls. So one student of mine has been working on these FLA11 and FLA12 and trying to understand really what their role is in this mechanical stress sensing. Okay, so he generated some overexpression lines. So these are under their own promoters. When we do it in 35S, doesn't give any phenotype, but when we do it specifically when they're expressed in secondary walls, you overexpress these and you get very stunted small plants. And these are expressed, both FLA11 and FLA12, as I mentioned, are expressed in cells with secondary walls. So in these xylem vessels and xylem fibers, and these interfascicular fibers, which provide a lot of strength. Okay, so they seem to be involved in mechanical stress because you have impaired mechanical properties when you lack these FLAs, and they're expressed in secondary walls. So do they regulate cell walls? Just before I go into that, let's have a look at the responses of plants to these compression and bending stresses and what the response in the cell walls are. So Yingxuan did an experiment where he put a weight on the stem to create this compression stress, so stress like weight coming directly down from the plant. And in that case, he saw that you get a thickening of these interfascicular fiber cells, so these ones creating this strength around the stem. And when you get bending, you get a different, you don't get the same thickening of these intervesicular fiber cells, but you do get changes in wall composition. So in the compression stress, you get more lignin, and in the bending stress, you get more cellulose. And what do you notice when you have these overexpressed in FLA11 lines, they get these thicker fibers as if they're under compression stress. So they get thickening of these intervesicular fiber walls. So here they get this additional sort of extended S3 layer that's much thicker than the equivalent in wild type. And he could quantify this. He also saw that the length of these fibers was much shorter, which is probably related to that thickening of that wall. Again, just because I've been talking for a while, I've got another poll for you. So here we've got fiber length of very little, about 800 microliters, but what is the longest plant cell that is possible? Thanks, Rebecca. How are we going? Woohoo! A root hair cell. Oh, it is the rami fiber, which can reach up to 15 centimeters. How incredible is that? 15 centimeters. Yes, root hair cells can get, like any elongation that a plant cell can do is incredible compared to those measly animal cells. 15 centimeters, incredible. So these are really, really strong. They can elongate because of their incredible cell walls. Anyway, just thought I'd throw that in there. Okay, so not only did they thicken these intervesicular fiber cells, but the composition was changed. So I mentioned before that you get more lignin when you get these compressive stress, but not only that, they had more lignin, but it was of a different composition. So when we looked at this, so there's different monomers of lignin, don't worry too much about this, but we saw that the G to S ratio of the lignin in these cell walls of the fibers, and well, actually the xylem vessels as well, was altered compared to what you would normally see in wild type. So FLA11 seems to be able to regulate not only the thickness of these walls, but also the composition. And it did this, we wanted to see when it was able to respond. So I said, potentially it's involved in these cell wall sensing pathways. So what is it doing? It's able to trigger this secondary cell wall synthesis early. So in the wild type stem that I'm showing here, this is a section through at that midpoint there, and you don't see any UV signal which would indicate secondary wall formation because the UV lignin can be seen with the UV filter. But when you look at the equivalent growth stage of the FLA11, they've initiated their secondary walls much earlier. So they really look like they're responding earlier than they should be in initiating this secondary wall formation. So we want to see if that is dependent on some of these transcription factor pathways. Sorry, before I go into that, we could see that, yeah, with transcriptional profiling, that was really, really clear as well, that these are initiating that secondary wall development. This seems to be the major responses in these plants, but also stress response was one of the major groups of genes that were upregulated in these plants. So we did, Yingchuan did a cross with the NST1, NST3 mutants that regulate that intervesicular fiber development. And we got a phenotype where these, the mutants crossed, the OE FLA11 expression lines crossed with these, also lacked the fiber, suggesting that the FLA11 is really dependent on these NST1, NST3 in the fiber cells. I'll just take note here that NST1 and 3 don't regulate xylem vessels, so these plants were still able to form xylem vessels, and that's likely why they're still short and stunted. Okay, so looks like FLA11 can regulate cell walls. Is it located at the right place? Well, as I said, these are really fascinating proteins, or glycoproteins, because they have all these different domains. So they have that vesicular domains, like I said, which have been implicated in protein-protein interactions. They have these glycans, which could make glycan interactions or glycan-protein interactions. They also have a GPI anchor, which could put them into particular lipid microdomains and co-regulate them with other, for example, receptor-like kinases. Just, yeah, they're fantastic. But we wanted to see where they actually are. Okay, so they should be located, because of this GPI anchor, at the plasma membrane. And so we checked both FLA11 and FLA12. And FLA12, you can see, does locate to the plasma membrane, whereas FLA11, sometimes it's at the plasma membrane, sometimes, and mostly though, it's in the wall, looking like it is cleaved from that plasma membrane and deposited into the wall. Okay, so there seems to be a bit of a, the FLA11 and FLA12 for a long time have sort of been lumped together. And I said FLAs flippantly, there's a lot of them, and they're very different. So we did a bioinformatic analysis. I say we, it was Thomas Shafee, who's a fantastic bioinformatician, who drilled down into more detail about what is the really distinct differences between these FLAs, because it seems like there's subtle differences in their structure are really, really important for how they might function. And so even if we look at FLA11 and 12 here, they're very similar. But there is some subtle differences in terms of their glycan sites, for example, and it appears their ability to be cleaved on that GPI anchor as well. And so Yingxuan has done a fantastic job really dissecting up the FLA11 and FLA12 proteins to see which are the really functional domains and which contribute to this overexpression phenotype that we're seeing. So he's generated all these mutant variants and looked to see where you get a similar phenotype to the overexpression line, or where you restore it back to a wild type like phenotype, suggesting that that's a really important domain that is needed for this phenotype. And what he did notice that was that the GPI anchor seems to be really, really important for that, because if you remove it, you no longer get this overexpression phenotype, and also the structure of that vesicular domain. What was interesting though was that when he looked at some other regions, so a surface B domain that could be involved in protein-protein interactions, as well as the AG2, which is the glycosylated region, you got almost a more severe phenotype, again indicating that these might be important regulatory regions. Okay, he's done a whole lot more in terms of swapping domains between FLA11 and 12, and it's, yeah, really interesting, so watch this space. Okay, so we've got a bit of a model where we think that FLA11 responds to this compression stress and is involved, therefore changing the cell wall so that these plants can maintain integrity as their weight builds up. And that changes largely the lignin. I didn't show it a lot here, but Yingxuan has evidence to suggest that they do actually work differently, and that FLA12 is more likely to respond to tension stresses rather than compression stress, because it seems to regulate cellulose. Okay, so there's lots of associated projects with this. So working in the Medicinal Agriculture Hub, looking at hemp fibre development and flowering. With Daisuke Takahashi in Japan, we're looking at the role of AGPs in cold tolerance and flowers as well. Also in salt tolerance. With Birgit Klassen in Kiel, tree biomechanics. This is a collaboration with Sean Mansfield and Weiting in China, and also rice stem strength in with Yua in Kaz in China as well. Okay, there's lots and lots of people who've been involved in this, and particularly I want to mention Lazar, Yingxuan, and Thomas Shafee, who've really contributed a lot to this project. Okay, that's me. I'm going to stop sharing. And if anyone has any questions.
So if you have some questions, can you type them in the Q&A box, please?
While we're waiting for people to type, maybe I can ask a question first as I can. So how do you think the FLAs are really sensing that tension? Do you think that there is some, like some of those leucine-rich repeats that you see, there's some actual sensing mechanical stress through the glycans? I know it's a bit hand-wavy at this point, but would you have any other ideas?
Yeah, no, I think they're going to be part of a complex that involves receptor-like kinases as well as some link to the cell wall. I think, and somewhere, I think there's going to be some phospholipases involved in removing that FLA11 also from the membrane, and whether or not, or well, whether that's really being pulled out or cleaved, I'm not sure. But yeah, there has, I mean, the FLAs themselves don't have any signaling capacity that we know about. They can interact with lots and lots of different things, but they need some kind of partner. And we have, Yingxuan's done a bit of work looking at wall-associated kinases. So there's a potential that interacts with one of those, but that's really where we want to go next, is finding those partners.
Okay, lots of questions, which is good. Phil, I don't know if Phil's able to unmute himself and ask his question, or?
No, Jenny,
no.
You'll have to read, you can read it out.
Sorry.
That's
so
okay.
Can FLA11 overexpression improve yields, like stomping on crops? From Phil Brewer.
I think that, yeah, good question. So that's really what we want to start to look at. Can we get a nice balance between that stem strength? I mean, you've got to be a bit careful with brittleness as well. So that can be a real problem. You don't want things that are really, really strong but really brittle, so they break over. But essentially, yes, we want to create crops that have a really nice strong stem that is resilient to some of these stresses that they might experience, whether it's people stomping on them or it's storms. We know that we're going to get more unpredictable climates in future. So yeah, that's really where we're aiming for.
Question from Matt Tucker. Not the tallest person in the world. Hi Kim, has anyone ever tried a genetic suppressor screen in the FLA overexpression lines to find mutations that rescue the phenotype? Yeah,
good. Not yet. Yeah. So, I mean, Yingxuan's only really just finished all those FLA overexpression. I think what's interesting I didn't show is that you can overexpress FLA12, no phenotype. So it's really FLA11 seems to be the dominant FLA for some reason. It's really specific. Whether or not that's just because it's regulating lignin rather than cellulose, I don't know. Yeah, really good suggestion. We'd like to go down that line. As I said, he's done heaps of those mutant variants now, and also swapping regions with FLA12. So we have a really good idea that the AG2 and GPI together seem to be really critical for its function. So that would be really, really nice to explore in future. But as for the pathways, yes, that's, yeah, support my next grant application and then I could work on it.
Question here from Scott. Do the FLA lines or other genotypes with modified secondary cell wall formation modify the vascular development in the stem? Yeah,
good question. So the FLA11 does also modify the vessel cells, as well as the intervesicular fiber cells. So, and FLA12 also is likely to be involved in the vascular tissues. And what's interesting about that one as well is that FLA12 seems to be broader. So when you overexpress FLA11 under the FLA12 promoter, you get defects in a lot of other tissues, not just the stem. And that's most likely because of changes in that vascular development in leaves and roots and sepals and things like that. So yes, and there's also, obviously, there's 21 FLAs, so there's more complexity here. So there's going to be a fine-tuning based on the regulation of those FLAs.
And so do you think that's why the 35S overexpression doesn't get that?
Yeah, from what we can tell, 35S doesn't really express well in secondary walls, cells with secondary walls. So you have to use a secondary wall specific promoter to see those effects. So I think therefore they're likely their interacting partners are also specifically regulated in those cell types as well.
Yeah. And so Phil, kind of following up on any effect of FLA11 on in roots, or just in general, like how do those root phenotypes compare? Yeah, so you do see a little bit of shorter roots in the FLA11 overexpressor because it initiates secondary wall development earlier also in roots. So it's not exclusive to stems, it's predominantly expressed in stems, but there's a little bit of expression in secondary, and all pretty much cells that make secondary walls. And so, yeah, you see a little bit of change in roots as well. I think that's anyone else got any burning questions? I think thank Kim for a fantastic talk. I really, really enjoyed that.
Thank you for having me.
Coming back a second time, and so yeah, wonderful.
Hopefully I can come and see you all sometime soon. Although you're still closing your borders to us.
It's not long, I reckon. Right. Thank you very much.
Thanks so much.
Thanks, Kim.